for physiology, we'll be discussing about neuromuscular and synaptic transmission. So first, we'll be identifying some terms. So for ion channels, these are integral proteins that may be open or closed, and this is a conductance of a channel. The channel is open, then there's a higher permeability. And these ion channels are controlled by gates. There are two types of gates, the voltage-gated channel and the ligand-gated channels. So in the voltage-gated channels, this are the membrane potential and activation or inactivation gate of sodium channel. And for the ligand-gated channel, where the hormones, second messenger, and neurotransmitters and nicotinic receptor for acetylcholine can be found. And we also have here the two potentials, the diffusion potential versus the equilibrium potential. So for the diffusion potential, this is a potential difference generated across the membrane because of concentration difference of an ion. So for the size of the diffusion potential, it depends on the size of the concentration gradients. And the sign of diffusion potentials can be either ion is positive or negatively charged. And the equilibrium potential, also known as Nernst potential, is the diffusion potential exactly balanced or opposed the tendency for diffusion down a concentration difference. So here are the equations. So equilibrium potential in millivolts is equals to negative 2.3 times RT over ZF log rho intracellular concentration in millimeter and extracellular concentration in millimeter. So the Z here is the charge on ion, whereas in there's a positive 1 for sodium, positive 2 for calcium, and negative 1 for chloride. And the equilibrium potential in millivolts in nerves and muscles are stated in the following. So the sodium have positive 65 millivolts, the calcium have positive 120 millivolts, and the potassium is negatively charged 85 millivolts, and chloride is negative 85 millivolts. Same with the potassium. Here we have the resting membrane potential. So this potential is exhibited by all cells, and it refers to intracellular charge, which is negatively charged with negative nerve RMP. So for the neurons or large neurons, there is negative 70 millivolts RMP, and the RMP for cardiac muscle is negative 90 millivolts. And these are caused by Nernst potential for sodium and potassium, and also for the potassium leak channel and sodium potassium ATPase pump. So for the action potential, so we can only see this on the excitable cells. They are only those who have action potentials, can be seen in all types of muscles, the skeletal, smooth muscles, and cardiac muscles, as well as in the neurons. So action potential have the on and off signals that propagates, and it has depolarization followed by repolarization. So the characteristic of action potentials are it is stereotypical size and shape, and the propagation, it means that it spread like electricities, and convection velocity is increased by fiber size. So the larger the nerve fiber, the smaller the internal resistance. And the conduction velocity is also increased by the nerve diameter. So and also in the myelination. So the myelin serves as an insulator, and the action potential is regenerated in the nodes of Ranvier. Nodes of Ranvier is an unmyelinated portion on the axon, and it contains the highest concentration of the sodium channel per square meter of the cell membrane. And the action potential have all or non-response. So when the threshold is reached, the action potential is inevitable. Here, let's discuss the parts of the neurons. We have nucleus, dendrite, cell body, or also known as soma, initial segment of axon, nodes of Ranvier, and the terminal botons. So the nucleus, this control and regulates cellular activities, and it carries the genes that is used in the production of the cellular proteins. And the dendrites, here we can see the neurotransmitter receptors, and in the cell body or the soma, where the organelles and nucleus is seen, and the initial segment of the axon, where the action potential starts, also known as the axon hillock. And we also have the nodes of Ranvier. This is the unmyelinated portion of the axon, as what we discussed a while ago. This is where the action potential regenerates. And the terminal botons, when the action potential reach the terminal botons, the voltage-gated calcium channel will open, and the calcium will enter, and there will be an increase in intracellular calcium. So there will be an exocytosis of the neurotransmitter. So we also have what we call saltatory conduction. This is the conduction across the nodes of Reinvier, and it makes transmission signal faster. It enables to regenerate action potential in each nodes of Reinvier. Now let's discuss some neurotransmitters, which includes acetylcholine, norepinephrine, epinephrine, dopamine, serotonin, nitric oxide, glycine, GABA, glutamate, and opioid peptide, as well as the glutamate and substance P. So first is the acetylcholine. So this is created by choline acetyltransferase from acetyl-CoA and choline. And this is degraded first by the acetylcholinesterase to acetate and choline before the reuptake, and it opens the sodium-potassium pump, depolarizes the muscle and plate, and it acts as a ligand-gated channel, so where the sodium goes in and potassium goes out. And this acetylcholine triggers the REM sleep, and this neurotransmitter is decreased in Huntington's dementia and Alzheimer's dementia. And we also have myasthenia gravis, where this condition is caused by antibodies to acetylcholine receptor, and this presents skeletal muscle weakness, fatigability, and increase in acetylcholine receptor. So that for the treatment of the myasthenia gravis, it's acetylcholine esterase inhibitor or neostigmine. Another neurotransmitter is the norepinephrine. So it is found in leukocortical use of the pons, and it serves as neuromodulator in the CNS and neurotransmitter in the PNS, and it is synthesized inside the synaptic vesicles. It is the primary neurotransmitter from postganglionic sympathetic neurons. So remember that norepi is the primary neurotransmitter in the postganglionic sympathetic neurons, and it is responsible for arousal or wakefulness. Another neurotransmitter is the epinephrine. It is secreted mainly by the adrenal medulla, and it has a greater beta-2 action, and it is given to status asthmaticus. Another neurotransmitter is the dopamine. It is secreted in substantia nigra and also in hypothalamus in a prolactin inhibitory hormone. So it inhibits prolactin if it's from hypothalamus. Remember that dopamine inhibits prolactin. And there are two receptors of the dopamine, the D1 receptor and the D2 receptor. So for the D1 receptor, it activates the adenyl cyclase using the GS protein. The GS protein activates the adenyl cyclase. So that's by the D1 receptor. However, if it is to inhibit the adenylate cyclase, we'll be using D2 receptor by GI protein. Yeah, so we have GS protein to activate and GI protein to inhibit. So I inhibit, S or stimulate or activate. So D1 is to activate the adenyl cyclase, and D2 to inhibit the adenylate cyclase using G1 protein. So in what conditions that can we see there will be a decrease in in dopamine level? It can be seen in Parkinson's disease. And remember the symptoms of Parkinson's disease includes the mnemonic trap, TRAP. So there will be tremors, rigidity, akinesia, and postural inability. And where can we see an increased level of D2 receptor in schizophrenic patient? So another neurotransmitter is the serotonin. So it is also known as the feel-good substance, and serotonin is low in patients with depression, and it is found in the median wrap of the brainstem. And serotonin is from the tryptophan converted to melatonin. Another neurotransmitter is the nitric oxide, and the nitric oxide synthase convert the arginine to citrulline and then nitric oxide. It is classified as an inhibitory, non-adrenergic, non-cholinergic, and it is a permanent gas, an inhibitory neurotransmitter, and it serves as vasodilator. So we also have here the glycine. Glycine is the main inhibitory neurotransmitter in the spinal cord, and it increases the chloride influx. However, in the brain, the main inhibitory neurotransmitter is the GABA, and it also increases the chloride influx using the GABA-A, or it increases the potassium efflux using GABA-B. And this neurotransmitter, the GABA, decreases the anxiety level. And the GABA A synthesized from glutamate by glutamate decarboxylase. So if the GABA is the main inhibitory neurotransmitter in the brain, the glutamate is the main excitatory neurotransmitter in the brain, and it activates the NMDA receptor. Another thing that we can find is the opioid peptide. It inhibits the neuron in the brain, and it involves in pain perception. So that's opioid peptide. This includes the enkephalin, endorphins, dernophins, except morphine. This is exogens. And for the trivia, we have here the fast pain and slow pain. Fast pain is an acute electric pain, and it uses type A delta nerve fiber that the neurotransmitter that's responsible for this one is the glutamate for the fast pain. However, for the slow pain, it uses type C nerve fiber, and the responsible here is the substance P. Okay. So another trivia. Let's remember the phenylalanine derivatives and the tryptophan derivatives. So our mnemonics for the phenylalanine derivatives is that pare, true love does not exist. So tyrosine to L-dopa to dopamine to non-epinephrine to epinephrine. So that's the phenylalanine derivatives. And for the tryptophan derivatives, these are the tripmoshano. So tryptophan form the melatonin to be serotonin and then niacin. So that's all for today.